Morning.
I turned it off so
Okay.
people wouldn't hear me.
Okay, good morning. So my name is Ramesh Goel. I'm professor civil and environmental engineering at University of Utah. So this is a bit different from other excellent presentations about June sucker recovery and carp and algae filtration. This is more fundamental, and then going to demonstrate the effect or the need for connected community type of effort when we are dealing with such a big eutrophication problem. So occurrence of harmful algal blooms is just not a local problem. It's everywhere, not only in the different parts of the United States, but around the globe. And the reason we care about cyanobacteria, we start with the hierarchy phytoplankton, and then under phytoplanktons are algae, and in algae then they are blue-green algae, and they are blue because they have this pigment phycocyanin under light gives you blue sign. And these are the one which we most care about. Apart from heavy eutrophication, of course, that other phytoplankton and algae can generate biomass and which can be degraded and consume DO. That is one aspect, but from health point of view, these blue-green algae or cyanobacteria, which generate the secondary metabolites, what we call cyanotoxins, are the one which are dangerous and we care about from health point of view. And there are primarily these three types of toxins that generate hepatotoxins, neurotoxins, and dermatotoxins. And we have seen some in the past that some people reported, rashes on the skin. Perhaps those are dermatotoxins. They're not exclusive. That who generates what, but there are some specifications and classifications, but what toxins are generated by which cyanobacteria. So types of, when we talk about even cyanobacteria, there are two types of cyanobacteria from their physical appearance and then their where they thrive. One is, of course, what we see in Utah Lake are planktonic cyanobacteria. They float, and not only they float through the water column because they have air vacuoles, so they go up and down. And something you see in the surface, most of the time you see like algae and cyanobacteria are floating in the water column because they compensate their nutrient deficiency by floating in the water column using their buoyancy force. So they're pretty smart creatures. The other type of cyanobacteria are benthic. They do form biofilm, thick biofilms, and we have seen several episodes of these very toxic cyanobacteria in Utah. For example, I don't know if you heard in 2020, Virgin River, a dog died actually. He just walked right before Narrows start, the dog just walked into and swallowed some of the mats. And then in 20 minutes, the dog died because they were neurotoxins. It was 2020 and 2021, similar episodes were observed. So we started sampling in Giant National Park also. We have pretty good data with a network of researchers from California, and it's pretty severe. And they tend to occur in shallow streams, of course, and form thick mats. But for our attention, we in this symposium mostly care about planktonic bacteria. So just a history how it evolved in 1970. You see, conventionally phosphorus was regarded as the nutrient, long-term limiting nutrient, fresh water, and for marine type of environment, then nitrogen was limiting. But the paradigm is slowly changing. The importance of nitrogen was noted as the bloom. Non-disostome is non-fixing microcystine and in rich surfaces. They produce enriched toxins, hepatotoxins harmful. And then slowly nowadays, what we see is a reduction of N-favored cyanobacteria community towards N-fixation cyanobacteria. So they can, because they are heterosis, and these N-fixing cyanobacteria can fix nitrogen, basically convert aerial nitrogen gas into ammonium and then use. So not only the cyanobacteria, these N-fixing can fix, but they can provide nitrogen to other non-nitrogen fixing cyanobacteria and other bacteria also in the system. So these are two nutrients of concern. Of course, there are many other macronutrients. We talk about silicate or we talk about other things. But when it comes to nutrient dynamics, nutrient paradigm, nitrogen and phosphorus are the two which are important from eutrophication, from harmful algal bloom point of view. So why I presented those cycles is phosphorus is pretty tricky element in the sense that it's just driven by redox chemistry, but nitrogen on the other hand is driven by geochemical chemistry, biogeochemical reactions. So you have ammonia, it's oxidized to nitrate or nitrite by bacteria, and the nitrate and nitrite are reduced to nitrogen gas by bacteria. On the other hand, phosphorus, when it comes into the system, unless there are chemical precipitations or it is flowing out of the system, so it's kind of a conservative, very conservative element. So dealing with phosphorus, of course, and evaluating its fate and transport is always challenging. But having said that, both cycle, whether nitrogen cycle or phosphorus cycle, we have to look at all components of these two cycles. So when it comes to nitrogen cycle, for example, we tend to focus on cyanobacteria, how much nitrogen is there, what is that CNP ratio, which is red fill ratio, typically been used for decades now. But one thing we forget that there are other bacteria plankton, that means non-cyanobacteria present in situ, which compete with cyanobacteria for nitrogen and phosphorus, especially for nitrogen. So something which was nitrogen, not nitrogen limited, becomes nitrogen limited because there are other bacteria plankton present. They can denitrify, they can oxidize nitrogen. And likewise, if you look at phosphorus, there's so much going on between sediments and the water column. There's atmospheric deposition, and then there are point and non-point sources bringing phosphorus, and then the phosphorus is deposited in sediments, and that is released under conditions. Even the different forms of phosphorus, loosely bound phosphorus, chemically bound phosphorus, the phosphorus which can be released or uptake because of redox potential, pH changes, for example. So this holistic picture we need to consider for any nutrient management type of study. So changing paradigm, there is a lack of scientific consensus on whether lake eutrophication should be managed by controlling N or P alone. Still, there are like millions of papers have come out over the last 4 decades, and still there is this confusion that how do we control? And this is not because we lack scientific understanding, it's because the dynamic nature of any water body shifts from N limitation to P limitation and vice versa during a bloom. So what we tend to focus is at the onset of bloom. When HAB, harmful algal bloom, occurs, then we see that, the body is water, it's nitrogen limited, and that's what caused the bloom to occur. So the phosphorus is the culprit, or the other way around. But what happens during a bloom, when the bloom takes place and then it peaks and then it subsides, there are changes in nutrient concentration, and I will show in next slide. And then those changes can drive microbial ecology, cyanobacter ecology during the bloom also. The bloom can last 1 month, the bloom can last 3 months, depending upon what kind of cyanobacteria are flourishing, depending upon their capability to undergo this stress management. The cyanobacteria, they do go under stress management for nutrients. And they are smart, they use their genetic tools to combat stresses in the water. Recently, eutrophication has been accompanied by a shift in stoichiometry. Of course, this is standard, red fill ratio is standard, but people have often found that they see harmful algal bloom formation or eutrophication at ratio other than red fill ratio. They're frequent, there are a lot of literature available now. So there is something going on. Does that mean we don't follow red fill? Yes, it is still the standard, and it gives us a lot of information. But in some of the typical environment, people have found that the red fill ratio doesn't follow, or the lake eutrophication doesn't follow red fill ratio. Nutrient uptake, this is same as my third bullet point, and cyanobacteria may be less vulnerable to neutral limitation than we thought. As I stated, there are cyanobacteria, Virgin River, when we measure nitrogen and phosphorus is non-detectable most of the time. And still we see thick mats of this genera, what we call microcoleus. This is like black, you will see slimy black stuff. Just if you see slimy black stuff in Virgin River, stay away. It's a toxic microcoleus actually. And they just thrive. There's no phosphorus and they thrive. And they produce these anotoxins, NAC, and there's a lot more going on there also. So they use their smart feature, genetic feature, to combat with these nitrogen and phosphorus limitations when these nutrients are depleted or very limited. So as I stated in my previous discussion that this is using metagenomic, the advanced genetic tools in my lab, where we show that what happens is during the bloom period started, we see this picocyanobacteria, the Cenococcus, now we start seeing Synobium also, and then stage 2, it becomes nitrogen limited. They all consume nitrogen. It becomes nitrogen. Then you see these nitrogen fixtures, Dolichospermum and Phenizomenon, they flourish. And then what happens is stage 2 is P limited, stage 3, then you see these N fixtures provided N. These are N fixtures, they provided N. Now you have these planktothrix and all these other toxic cyanobacteria flourishing. This is based on real genomic tools. And this is exactly what happens in lakes when there's bloom. So rather than just focusing on one point of bloom, it is very important to focus on a complete cycle of bloom and what happens, how different cyanobacteria they thrive, what is their ecology. This is an example of what happened. This exactly the previous figure came from this and some metagenome. And you see, this is more important. This is at phylum level. And when you zoom more into, then you get this genus level diagrams. And you see right there, the yellows are Synobium, and then blue is Phenizomenon. But you see other bars. These are like toxic cyanobacteria based on this. And this is during a complete bloom cycle. So this is interesting, but a bit complicated. But what I want to emphasize is what we did is during a bloom, you just don't only look at a small segment of DNA. You recovered the whole genome of cyanobacteria. Why is that important? When you recover the whole genome of cyanobacteria, then and you have genome of other cyanobacteria also. So now you have genomes of several toxic cyanobacteria, and then you compare those genomes. And by comparing those genomes, you can tell that, hey, this cyanobacteria needs help from this cyanobacteria to carry out this metabolism. So that's exactly what we were doing. So these are genomes of cyanobacteria, metagenomes recovered from Utah Lake during a bloom cycle. And we can see that they were doing nitrogen fixation, as they are known as nitrogen fixations, or Dolichospermum and Phenizomenon. And then on the other hand, during nitrogen limitation, they were using their spatial attribute, nitrogen regulation, to get nitrogen through nitrate, and likewise right here. And then they're coping up with nitrogen stress. So these kind of information, this was published in ESNT recently. So this kind of information becomes very important when we want to answer question like, hey, this lake system or this freshwater system is unlimited. Why these are flourishing? What I do need to do? So sometimes that CNP ratio doesn't work. And this is another thing I mentioned about nitrification. So these are bacteria plankton. That means non-cyanobacteria. These are cyanobacteria. So as you see, denitrification was performed by other bacteria. That means it's just not cyanobacteria consuming nitrogen. There are these bacteria plankton, they are consuming nitrogen, converting nitrogen into nitrogen gas, making the whole system nitrogen limited. So these considerations, so looking into the synergy between cyanobacteria and other bacteria becomes very important. So I just have 1 minute, but humans obtain, management point of view, we obtained benefits from outstanding services from ecosystem. Freshwater systems, lake connect several interconnections, as the previous presenter showed that the people look so happy, you have park and you have this recreational area. In general, around the globe, decision making related to the reduction of eutrophication, mostly unidirectional. A lack of connectivity and cooperation between community members and the need for a more robust data-driven understanding. So this is an example is when we pose technical questions, we have to have social questions. And these technical questions and social questions, they need to be integrated together. They cannot be just stand alone and then we do. And that's where the disconnect is. We need to integrate what are my social questions, what are my technical questions. So just an example of this study, this is like an example of connected community. The study was initiated by Tipanoga Special Silver District. Rich is there. Rich, thank you so much. We installed 9 limnocorals in Utah Lake, and these limnocorals were in like 7 to 9 meters in diameter. So in this room, you can probably put 3 of them, that's it. So there were 9. So we all sampled, they were spiked with 2 different concentrations of phosphorus at 2 different point of time, in July and September, and they were open lake water sampling also. So 9 plus 4, 14. So the lot of like 30 parameters were monitored. A huge team of people, as you see, Jacob Engineering, private, and then TSSD and BYU, everybody were involved in this whole sampling. And you see, what I like about the connected community. This is connected community. You cannot stand in. They were just shaking, windy. It's sometimes you just full like feel like falling and swimming in these limnocorals. But they're big, and you see how we're mixing when we put concentrated stock solution of phosphate. That was our way of mixing because they're huge, 300,000 liter or something. That's big. So big effort, but it was all community-led. So these are data from all. What we found is these were their microcysty, and then some toxin provision were there, but they were not different from the open lake waters. That means the limnocorals were spiked with phosphorus. The results were not very different from open lake water where we did not spike with phosphorus. So among many conclusions, that was like main conclusion. There is a big report being posted, prepared by folks. It's like 200 pages report. Eventually it will be posted. That contains invertebrate, fish data, and other data also. So just my last slide, that what happens is, in a connected community, so you have driving factors, real estate, for example, local businesses, and tourism. We don't consider. We have to convey this information to local residents. Say, hey, improving lake quality means these things, and these things going to help local quality. If I tell, go to a resident local, I don't blame people. Say, I'm going to raise your sewer bill by $30. No, I'm not going to raise. But if we convey this same information on social grounds, on economical ground, that this is how it's going to help you, then it makes sense to residents and citizens. Then they will accept, they will come together, and that oftentimes doesn't happen. And then in terms of interventions, then what we can propose like free shops. So this is what I call bottom-up approach, and then green infrastructure, for example, bioretention pond, controlled fertilizer use. So that's it. In summary, formation is a persistence problem. So the conclusions are there. Thank you so much.